Hello, everyone. Welcome to our session today. And we're going to be talking about what's new in research with Dr. Jaehyuk Choi. It's really my delight and honor to introduce you to Dr. Choi for this more advanced track that we are going to be talking about what's new in research. And for those of you who do not know Dr. Choi, he's an associate professor of dermatology and biochemistry and molecular genetics. Whoosh, that's a mouthful. And medical director of the extracorporeal photophoresis unit and the director of precision immunotherapy for the Lurie Cancer Center at Northwestern in Chicago. He's published more than 50 manuscripts in high impact journals and has been acknowledged with multiple honors, including the NIH New Innovator Award. And more importantly, or at least for us personally, Dr. Choi is a member of our Research Advisory Council and has been our Scientific Review Committee chair for the last several years. And his goal in the laboratory and in the clinic is to improve the ways we treat patients with skin lymphomas. So he really is the perfect person to talk to us today about what's new in research, and we look forward to hearing from you. Dr. Choi, thank you so much for joining us, and I'm going to turn it over to you from here.
Great. Thank you so much for having me. I'm really excited to talk about research in cutaneous lymphomas. It's been an amazing journey for all of us who are interested in improving the way that patients are diagnosed, staged, and treated because of the real advancements in our understanding of the disease and also how we are thinking about implementing those in the clinic. And I just wanted to say that I am actually a physician scientist, so I see patients in clinic. And in addition, we study cutaneous lymphomas in the laboratory where I run an NIH-funded laboratory with a number of graduate students, postdocs, physicians, and junior faculty members who are all unified in their quest to make your journey better. And so with that, I'd like to get started, and I'm going to have a few anecdotes from our own personal experience, but we're also going to talk about the field, and I would try to give as broad a vision as we can. I will say that I pegged this talk to be something similar to a talk I gave for Congress in 2017, but that may be below your level of sophistication, to be honest. So I'm happy to take questions at the end. But I will try to stay high level, but also really try to gear in towards the real advancements that you should be expecting to be translated into the clinic in the near future. So first, I just wanted to say is that, I just wanted to show this picture of one of our patients. And the reason I wanted to do this is just to remind us all what we're trying to fight for. And I think, a lot of struggles occur for our patients, and it really stems from even this picture. So this is a picture of mycosis fungoides. It's one of the types of cutaneous T-cell lymphomas. And one of the things that's really remarkable is always how it can always look like a rash, which leads to delays in diagnosis, potential delays in treatments, but also it befuddles, to be honest, the physicians and the scientists who are studying this disease. But I think we've made significant progress in how we're thinking about it, which will help us to understand, one, why it looks like a rash, which I'll be happy to discuss in questions if people have any questions about this, but also how we can really utilize modern technologies to better understand the disease and improve treatment outcomes. So I wanted to show this slide because I've shown this slide now for actually close to 10 years. And what's striking to me about this slide is actually still that it's still a lot of the way that we treat patients. We have patients and we do a very detailed workup. And this is especially true if you go to a major center that specializes in cutaneous lymphomas. The workup is highly detailed and extremely molecular and advanced. But nonetheless, we often decide treatments not based on what the individual patient has per se, but really based on our broad experience with the treatments themselves. So at Northwestern, for example, I can tell you that for a lot of our patients, we decide treatments really based on safety profiles because we don't have enough information to predict who's going to respond to individual treatments. And what I'm going to talk about today this was our dream from about 10 years ago, and I will be very excited to tell you that this is quickly becoming our reality. And the emphasis of my talk, which will be broad, is really talking about how we've now developed tools to be able to try to answer questions I get from clinic all the time. And one of them is, what is going to best work for me? And this is the goal of my research, as well as many investigators in the field who I'd be happy to acknowledge. But many of you know them because they're your treating physicians at your centers. And our goal really now is to really take advantage of the scientific revolution that's occurring in biology, and in order to help understand why people are different and how we can treat patients better. And so I actually hearken back to a lot of my patients and what they're trying to ask me. And I will say almost every new patient asks me these three questions. Why do I have this disease? What should I expect to happen? And what do I need to do? And my goal in the laboratory, as well as in the field, we're really trying to answer these three questions. And I can tell you, as having served on the Cutaneous Lymphoma Foundation Research Advisory Council, these are the three questions that we're also trying to answer with the foundation. And the reason why this is a little bit more difficult is because the disease is actually very heterogeneous, meaning, as you see here on the left, many different colors of people, that actually represents cutaneous lymphomas. They can be very different from patient to patient. The disease is relatively uncommon, meaning it's very difficult to actually enroll patients into a lot of clinical trials compared to other more common diseases. And really, we have to think about, as a field, how to collect the right types of data and how do you use this data to make clinical decisions. So again, a running theme in my presentation will be to try to think about, why do I have this disease? What should I expect to happen to me? And what should I do in the future for me? So this is the area where I'm going to use the schematic to describe really the efforts in the field, both in our laboratory as well as in the field. And the idea here is that, probably in the 80s, 90s, and early aughts, I would say that most patients actually looked like this, meaning that we really had a tough time understanding what makes the disease work and why the disease happens. But over the last 10 to 15 years, we've worked as a field very hard to try to utilize modern technologies to understand what drives diseases to be diseases. And utilizing these kind of cutting edge technologies, which I will mention briefly in the future, we're now able to paint people with a specific color, which really describes the different drivers of this disease. So let's just say this is the red kind of cutaneous lymphoma, and this is the green kind of cutaneous lymphoma. We're really getting a better understanding of the factors that make this person green and this person red. But our goal, which is really the beginning of our talk, is to really take this information and utilize it in the clinic, which will take more research. And so what we're working on now is, okay, we are starting to understand what makes people different. What are the colors that are the sources of differences between patient to patient? And can we utilize this information to generate, clinical trials and clinical algorithms so that each person can receive the treatment specifically for them? This will take in consideration what patients do if they didn't get treated, and also how they would respond to common FDA-approved therapies that we already have access to in the clinic, as well as one of our dreams, which is to identify new therapies specifically for subsets of patients for whom it'll benefit the most. So I wanted to put this cartoon together because this really goes through, I would say, the last 5 years in the field and the way that we're sort of being poised for the next steps. And I will talk about some of the future technologies in the future. But really what has happened is, through major syndicates across the world, we have really focused on patients, and we've enrolled patients into our studies. And then we do a number of molecular assays. And so you don't have to worry about these, what we're saying here. I'll talk about it again. But we have assays that can tell us about what cells are in your tumor, What does the DNA of the tumor look like? What does the RNA of lymphoma look like? And utilizing all this information, we can take these big data approaches, sort of the same types of big data approaches that are being used by companies like Google and Apple to help you do better searches or to do better, understanding of why things happen in the real world. We're trying to apply these algorithms to your clinical situation. And so our goal now is to be able to identify the molecular explanations for why the lymphomas occur. We're trying to see if this reflects clinically relevance to how you should be treated. And these kind of things have to be functionally validated in the laboratory as well as hopefully in prospective clinical trials. And so this is the approach that we and many others are taking to try to really weaponize the information that we're getting in the laboratory to better fight this, this disease. And so I'm going to talk about several big concepts, and I'm happy to go through them more. But in general, what I wanted to say was that lymphomas are derived from specialized immune cells, and these are T cells or B cells. You may have heard a little bit about them during the COVID pandemic because these are the types of cells that help you to fight off infections. And what I'll say is through, nearly miraculous means, we have many, many different types of immune cells in our bodies. And each of them is very specialized to help us fight off different types of infections. And so you can imagine we have antiviral, antibacterial T cells, or B cells, and each of these can have very specialized functions. And I reflect this here with the different colors that we have represented: the blue, the yellow, the red, the green, and the light green and blue. And what I want to say is that what we've really learned over the past 5 years, especially the past 2 years, is that each of these cells can actually become a lymphoma. And the source of the cell, the different type of its cell it's derived from, actually is probably one of the major reasons why patients are different from patient to patient. The second thing I wanted to say is that your cancers or lymphomas actually have their own genome. And so it turns out that we're all born with DNA. We inherit half of our DNA from mom and half of our DNA from dad. And this DNA is what we're born with and helps to guide us to see how tall we are, the color of our hair, etc. But it turns out the lymphoma cells themselves, they have their own DNA. That means the information you got from mom and dad has been corrupted. This DNA in the lymphoma cells can actually become mutated. And when they do, this is called a somatic mutation or a cancer-specific mutation. And these mutations actually drive the formation of lymphomas. And so because these mutations are an early event and what we call a driver event in lymphomas, understanding what these mutations are doing is really critical to understanding what is going on with the lymphomas in combination with the different cell types that we see. So this is complicated, but I just wanted to show it here because this is actually our model. And I'm going to try to break this down in different ways with the examples, but really I wanted to give you a sense of where we're going and how we're using this information to better understand the disease. And so what we have done as in my lab, as well as across the syndicate of investigators associated with the Cutaneous Lymphoma Foundation and across the world, is that what we have been able to do with increasing precision and robustness is to be able to identify the cell that is from which the lymphoma is derived. We've been cataloging all the mutations that are occurring in these samples, and we can now better understand with increased precision how these different combinations of mutations and the cell of origin are leading to specific disease. One thing here, which I don't want you to get bogged down with the terminology, these are called cytokines and homing molecules, is just to say, as I mentioned at the beginning, cutaneous lymphomas can look like a rash. And the reason that is because they often retain the intrinsic properties of the immune cells that actually cause rashes. And these molecules are also still expressed by the lymphoma cells. And that's why you get the disease the way that it looks, the way that it feels, and all the different aspects, even including itch, as well as others, which lead to the clinical phenotypes associated with your disease. And phenotypes means how you feel and what you look like. So I just wanted to give this as an example. This is a cartoon of an example where we have really now applied these very strong techniques to try to understand why patients may be different from one another. And so what we have found is that these patients, if you look in the left, this is a schematic of human skin. And what we have found is actually that the different types of T cells are different depending on what layer of skin they come from. And so if you look at the skin, this is a hair follicle, which is the hair that's on your arms. And you can see this is the sweat gland, and these are the muscles that become tighter when you have goosebumps. And you can have cells in the top layer of skin, the middle layer of skin, and the bottom layer of skin. And each of these things, even though they're all part of the skin, are actually slightly different. And what we didn't fully appreciate until the past year or so is that the different layers of skin actually have different types of T cells. And each of these different T cells actually lead to slightly different diseases. And being able to understand this, we're not only now able to try to think about how do we give precise clinical information based on the information that we have already and the treatments available to us now, but we are also utilizing this information to try to develop treatments very specifically for lymphomas that derive from the lower layers of skin, the middle layers of skin, or the top layers of skin. Through our work, I mean, again, the Cutaneous Lymphoma Foundation associate investigators, my lab, as well as many labs across the world, we have collectively been able to identify the different types of mutations that occur in the cancer genome. Again, these are the aberrations in the DNA that you originally inherit from mom and dad, but only occur in the lymphoma cells. And we've been able to utilize this to really have a really fine-tuned genetic map for all the different lymphomas that are occurring across different patients. And we're utilizing this information now to try to identify new treatments. And I will discuss some of these examples as we move forward. So this is an example where I just really wanted to say is that, you know, we have been able to work together with our colleagues to really look across the landscape. And so to many of you, these are really, you know, uncommon diseases as, you know, in terms of the population, but that doesn't make it any less meaningful for the patients. So our goal is to really take these different cutaneous lymphomas and really identify what makes them tick. And we think by understanding what makes them tick that we're really able to figure out, you know, specific therapies that are designed for patients with these diseases. And so here I just wanted to say is that we've really made dramatic disease progression in how we have aggressive B-cell lymphomas. And we've utilized this information to launch a clinical trial just for these patients. We've now utilized genetic information to identify patients for whom the B-cell lymphomas are never likely to pose a threat to their health, and trying to spare them any toxicity associated with imaging scans and also with possible overtreatment. And we've been using these approaches across many different diseases. Here I just talked about the gamma delta T-cell lymphomas. This is a specific type of T-cell, but it's also a type of disease that afflicts some patients with cutaneous lymphomas, as well as CD8 T-cell and CD4 T-cell lymphomas. Again, each of these represent 3 of the different types of immune cells. You could think of them as representing the red, blue, and green cells that we showed before, and that these are all the different types of lymphomas that can afflict each person. But to be honest, many of our treatments, clinical trials, are not designed for only one subtype of lymphoma. So we do not have specialized treatments right now for each of these individual therapy diseases. And our goal is to utilize information, which we have done now, to better understand why they're different and to utilize this information to try to design patient-specific clinical trials. Okay, so before I go there, I just wanted to say that this will boggle your mind, I hope, because it boggles my mind, is that we've now been able to take a patient workup, which was originally based on a pathologist review of the biopsy, as well as the clinician annotation of the skin exam. And now we've been able to take this and make this into a 3 billion base pair assay. So in this case, we're taking each of these samples. This is your lymphoma, someone's lymphoma. We're able to generate some of these RNA and DNA, and then we're able to see how it looks like across the 3 billion base pairs of the human genome. And this has been amazing because it's been able to help us to decide how patients are different. And I will say is, when we get this information, it comes out as sort of like this, which is what we call a heat map. But I'll give you the analogy here is that this has been very helpful, but actually, it's been very analogous to drinking a smoothie. And so when you drink a smoothie, you can tell that it has mango or strawberry or avocado, but sometimes it's difficult to tell all the different ingredients. Now, this information has really been helpful already. It's launched many preclinical data that hopefully will be brought into clinical trials very soon. But it actually, what we have been striving to do as a scientific field is to take these smoothies and figure out the different components to them. What fruits do they come from? And so the real advance in the past year have been really aligned with this process. So we now have these really amazing scientific tools where we're able to take a biopsy sample, make each of the samples go down to the individual cells. These are the cells that are in, each of these cells. So we can have all the different lymphoma cells separate. And now we can do RNA-seq or any kind of molecular assay to each of them individually. And utilizing this approach, we're now able to precisely measure how tumor cells are actually different within patients. And so before I was telling you that we're getting different colors of patients from one another, but now we're able to actually look microscopically and see the different molecular features that make one lymphoma cell different from another. And so my analogy here is that instead of having a smoothie where everything is blended together, you can now imagine we have almost close to a fruit plate or a charcuterie plate, because now we're able to see the relative contributions of the blueberries, strawberries, olives, and cheeses that really make up the different lymphomas. And the reason why this is so important is because many things that affect our patients are probably associated with individual cells that have gone rogue. We have many patients who do well with one therapy and then it stops working. And we all think as a field right now that when it stops working, it's because you had one cell, like here, maybe one fruit, that actually slightly different and resistant to treatment and allows you to become resistant to treatment. But if we know these differences in advance, we can design better therapies and therapeutic regimens that will help prevent you from becoming resistant to therapies. And this has been the most recent revolution in our field, which is ongoing, which is that we have been able to utilize as a field hundreds, if not close to 1,000 patients to utilize our idea of what makes patients different from each other, but that's at the smoothie level or the bulk RNA-seq level. Now we're able to do is individualize, get all the different cells from the different tumors and identify what makes them different from one another. And this is utilizing the special single cell approach, which takes advantage of world-class microfluidics as well as really sensitive genome sequencing efforts to identify how these patients are different. I can't tell you yet how we're going to use this information in the future, but this information will likely be really critical to really understanding at the single cell level how your disease is going to change during treatment and to really predict what will happen in the future. So hopefully I'll be able to help you think about why and how we are doing so great in our fight against cutaneous lymphoma. But where are we going to go from here? I've discussed with you a few things. These are the biomarkers. These are different pieces of data that are being available to the clinician to best help them to advise you what is the best therapy for you. This is a continued fight for us that we're continuously trying to do. I can tell you, as the director of photopheresis, the first question I get is always, is this going to work for me? And I can't say that now, but I will predict that in the next few years, we will be able to utilize all these really cutting edge technologies and our collective field efforts from our work, the Cutaneous Lymphoma Foundation work, which is run by their investigators, as well as funded by their research grants, to utilize this information to better understand who is going to respond. The second thing is that because the disease is relatively uncommon, we really need to identify the best agents for new future clinical trials. In some cases, like in more common diseases, it is routine practice to try clinical trials for almost every new agent. It is just not possible for disease that's uncommon like this. So we're going to continue to our fight by utilizing all the great research that's going across the world to be able to design better clinical trials. And hopefully I can give you an example of that to come. So what I want to do is, in the last part of my talk, is I really want to talk about clinical trials. So when I talk about what's new in research, for me, that really means new discovery of molecules that can be studied in the laboratory and brought into the patients through medications. But for a lot of patients, what they want to think about research, it's really about clinical trials. And so first, I want to talk about what clinical trials are like, just briefly, and how this kind of pipeline for new drugs occurs. But then I want to talk a little bit about something that we can do very quickly now, and as well as the landscape of clinical trials and where you can learn more about it for yourself. So first, I wanted to highlight again is that I've really focused a lot of my talk on discovery and preclinical efforts. And so in these areas, we're really interested in utilizing information from patient samples, utilizing cutting edge scientific approaches to really identify why the lymphoma occurs. And we think if we understand why the lymphoma occurs, we can design silver bullets that will be able to kill the lymphoma cells. Then this concept can be brought into the clinic with new therapeutics. They're used to in patients through different phases, phase 1, phase 2, and phase 3 clinical trials, to be able to show that these medications, which are really founded and grounded in science, can be utilized for the right patients. And our goal is to take this to FDA approval so that you can, and the clinicians and the patients can have access widely to these therapeutics, and they can be used exclusively on label for the patients for whom they will work the best. So with that in mind, I'm going to talk about one novel biomarker-driven trial that we have started in collaboration with some of our colleagues at Sloan Kettering, also at Northwestern with Joanne Guitart, as well as at the Dana-Farber Cancer Institute at Harvard. I'm going to then talk about two different classes or ways that we try to fight cutaneous lymphomas, and I will try to give you a landscape of what agents are in clinical trials now. And then I'm going to end a little bit about talking about research and why it's so important. So I want to say is, in some ways, the future is now. And this is something that we've dreamed about in our laboratory as well as in the clinic. We always dreamed that there'd be a way that you could find a person's disease, put it through a machine, identify what is driving that disease, then take off the shelf a therapy that's already available and give it to those patients. And this is actually occurring and being implemented as we speak. And so you don't have to worry about the specific subtype. This is a very uncommon subtype, even within cutaneous lymphomas. But we've actually found is that in these patients, by using this whole genome sequencing approach, where we sequence across the entire 3 billion base pairs of the patient tumors, we have found disease-defining mutations that pretty much only occur in these patients. These are called DNA fusions because they take your DNA, which are in different genes, and they mix them together to form this new chimeric hybrid gene that shares properties of gene A and properties of gene B. And we have found that in these lymphomas, that almost all of them have these fusion genes. We can now utilize this information to more precisely diagnose these patients because we know these kinases or these fusion products are actually critical for disease pathogenesis. What we have done working with our colleagues at MGH, Dana-Farber, Sloan Kettering, and here at Northwestern is that we know this fusion of gene A and B are really critical for disease pathogenesis. So we have been utilizing sequencing-based efforts with drug discovery to identify drugs that really work for these cells. And then now we've actually launched a clinical trial for patients with this disease, utilizing our efforts of sequencing to be able to identify, again, the gene mutations that occur specifically in these lymphomas, not in all cutaneous lymphomas, but this one particular subtype that make them very sensitive to this specific drugs. And so this is what I envision for the future, and many in the field envision for the future. We would like to do is have a patient come in, tell them exactly what's going on with their lymphomas, and then if needed, really be able to identify a specific, hopefully magic bullet that'll be geared towards targeting that specific patient's lymphoma, putting them through clinical trials that hopefully in the future, this therapy, if effective, will be utilized by all patients with this disease. Okay, so now if I step out more broadly, there are many, many different clinical trials that are available for our patients. And for here, I just gave a schematic. So in here, this different cell on the bottom is a cutaneous lymphoma cell. It presents antigen or can be recognized by T cells, which I show here in the aquamarine green-blue. And it also expresses proteins, which I show here in the light blue triangle, that can be recognized by different antibodies. And so I classify these different agents as immunotherapies. And so there are many cutting-edge immunotherapies designed for cutaneous lymphomas. And for here, I'm even only taking the therapies that are actively in clinical trials, and you can find them on the website clinicaltrials.gov. So to tell you a little bit about the future, which is almost like science fiction and how amazing it is to utilize technologies and engineering to really improve treatments for our patients. So number one, we have, not we, myself, but people in the field have developed T cells that they can engineer to specifically recognize and try to kill the cutaneous lymphoma cells. And so these are agents that are being utilized in clinical trials now, but if successful, may be broadly available for patients with cutaneous lymphomas. So that is taking your own immune cells for patients, engineering them to be slightly different so that they can actually recognize and kill the cutaneous lymphoma cells. We also have a number of antibodies. These are, which we may all know now because of COVID. These are the different circulating proteins that can block protein-protein interactions. So in these cases, these are proteins that can be utilized or antibodies to reawaken anti-tumor T cells. These are being studied in clinical trials as well. We also have new engineered antibodies that can actually bring the T cell to the lymphoma cell to try to kill them. And these are also in clinical trials. We have agents that are antibody-like reagents that bring almost like a nuclear warhead to the tumor cells to try to kill them. In this case, it's E777, as well as we have new combinations with FDA-approved therapies or antibodies that recognize proteins, in this case, the triangle expressed by lymphoma cells, that can be really utilized to target and selectively appoint your immune system to try to kill the lymphoma cells. Lastly, I wanted to show, and this is a little bit of high school to college biology, but we have this DNA at the top. This transforms into RNA, which then becomes proteins, and these lead to cancers. And this is what we call central dogma in biology, how the DNA that you inherit from mom and dad lead to different proteins, to why we're different from one another. And if you look at this, this is the central dogma that also applies to the cancer cell. And there are many different therapies that are used to target different aspects of this process. And so if you look at the top, we have these new molecules that can degrade or get rid of proteins that promote cancer by causing RNA to be made from DNA. We also have HDAC inhibitors, new ones being made that also target this process of DNA being made to RNA. And then we have new retinoids that are being tested in the clinic. We have these very fancy RNA molecules called antisense oligos. This was the subject of a Nobel Prize about 10 years ago. These can be used, again, to prevent formation of proteins that promote cancer. And then lastly, we even have new inhibitors of the proteins that promote cancer, in this case, an ITK inhibitor, as well as PI3K delta inhibitors that are in clinical trials for patients with cutaneous lymphomas. So I wanted to go back to the original slide, which really drives our research, and I think the mission of many people in the field to really answer for our patients. Why do I have it? What should I expect to happen for me? And what should I do? And I hope that I will be able to tell you now and in the future that we have a lot of hope, a lot of momentum, and a lot of energy to be able to answer these questions. We can identify with increasing precision what makes each person different from one another. We can even identify what makes each lymphoma cell different from one another. We can see why it's different in the skin and the blood, and we are utilizing this information with increasing rapidity, quickness, and with robustness to try to get the right patients the right therapies. And our example with the clinical trial that we're designing, or we're actually opened, is an incredible triumph of many, many different investigators, as well as many, many new technologies, as well as our patients themselves, who really helped us to understand how we can better understand the disease, because our goal is to treat and find new ways to treat these diseases. So where do we go from here? I just wanted to say is that actually we need more research, and all of you have done your part to help support this research, because a large part of the research actually comes from the NIH, which is supported by taxpayers in the United States. But we need more research, and this will continue to occur through efforts from the NIH, from different anti-cancer foundations, from the Cutaneous Lymphoma Foundation itself, as well as even from drug companies. And the reason why we need more research is because of three things. So number one is, want to say was we want to continue to make breakthroughs. And so in our breakthroughs, we really have to focus on trying to develop new therapies for these patients, because we are able to. And so we need to be able to apply this model of developing clinical trials for specific patients across the board so we can give patients the therapies that they best need, not overtreat them, not undertreat them, and help them to be able to have complete remission from their disease. I will say also that one of the key aspects of the research that we're trying to do is we also need the next generation of leaders. And so a lot of the new people who are coming through in the field will require training and rigorous training through doing research and scientific efforts to help them to not only be outstanding researchers in the laboratory, but also future leaders in clinical trials really have to understand the molecular basis of disease. And the last thing I want to say is that we really have to help our leaders to focus. And I will say is in the United States in particular, much of the efforts of physicians, physician scientists, and scientists are actually spent trying to write grants for more research. And what we would like to do is to really energize the discovery phase, just like discoveries for new treatments and vaccines for COVID, that by utilizing resources and people and focusing them on cutaneous lymphomas and the question at hand, we'll better be able to have leaders in the field focus on the questions at hand so we can spend more time doing the research and translating it into clinical care. So I'll skip that. So I just wanted to end by saying that the motivation for all the research for everyone in this field is the patients. And what I have here is an example of a patient that I saw when he was in rehab. And this is a picture taken actually by my daughter. And I promised the patient that we will do the best we can, both in the clinic and in the laboratory, to help him with his disease. And so he has kindly allowed me to show his picture because I have told him that he's going to continue to be the inspiration for what we do. And so even though we've given these very specific examples of research and how they can occur, our goal is always the same, and it's very broad and ambitious as a field. And our goal is to continue to try to cure CTCL and CBCL and all the different cutaneous lymphomas that we have. And it's always driven, I think, by the patients with patient material to design new treatments for our patients. So with that, I'd be happy to answer any questions, but I wanted to say again that it's really a pleasure to speak to all of you. Research is my passion, and I really want to improve patient care to be able to answer those questions for our patients in the clinic. And we will work together as a field with patients, physicians, and scientists and the Cutaneous Lymphoma Foundation to try to develop new ways to be able to treat patients to make sure that they get the best care that they deserve. Thank you very much.
Wow, Dr. Choi, thank you. That was fabulous. And I hope everyone feels as excited and hopeful as I do in listening. I mean, you know, I have the luxury again of listening to the scientific presentations at the different clinical meetings, but you kind of condensed everything that we've been hearing into an understandable format that is just totally amazing to me. How much has been learned in this very short window of time using new technologies, right? To really be able to pull apart the different cells within a lymphoma cell is just, wow. And to answer that question about, you know, the tagline in cutaneous lymphoma is always, if you've seen one patient, you've seen one patient, right? And then how do you treat that individual's disease iteration if you can't understand what it is, what their disease really is? So this is a huge step forward in being able to do that. So, I mean, I have a ton of questions, but I hope everybody will put their questions into the chat. I've got a couple of questions here for you. Let's see. I'm going to ask them, and then you can take them from here. From Joe, is the RNA sequencing, disease cell genome sequencing, readily available? And if no, where and how is it available? And I think that's a big question too, right? How does an individual gain access to this kind of diagnostic?
So I will say is that in the field of medicine, the use of RNA and DNA sequencing is increasingly being part of the clinical workup. I can't promise it for everybody, but there's a number of commercial entities and universities that will provide some of these services. What I will say is that my laboratory is trying to provide these services across potentially the world. So where we'd be able to provide this information for patients potentially in real time. It's not up and ready yet, but that is the vision that we have, is that we would like to be able to provide real-time information in digestible clinical information for both the physicians and the patients. So I would say is the three things I would ask is I would ask your clinician, because there are different access to these different types of information. Not everyone is going to be able to use that for you in particular in real time, because each of these studies takes some effort to really figure out how to crack the code of the lymphoma for each patient. But if you bear with us, we will continue to make more efforts to go forward. So in summary, physicians have access to ways to do this. There are also research studies to do this in an international way. And we're going to try to spearhead one ourselves. And, our goal is to be able to give you real-time information that you'll need. So your physician will have access to some of this types of technologies, but we will continue to try to bring more and more of them into the clinic as we go forward. What access you have depends on what access the physician has, because some of these tests are administered at your local hospitals, but some of them are also send-outs to different vendors, which are different based on where you're being seen. But in the end, we think that what will really drive this is to continue to apply these types of algorithms to patient samples before they're ready for clinical translation, so that we can understand what they'll do. And so I think I would continue to encourage all of you to participate, if possible, if you're a patient or a loved one of a patient, to these research efforts to be able to understand what makes their lymphoma click. Hope that answers your question. Yeah.
Yeah,
but
we're still, go ahead.
Oh, sorry. It just really depends on your hospital and these different vendors. So I will be happy to help if there's someone who doesn't have information, but I'm sure your physicians will have access to some of these technologies for clinical care. But we do need more of this information also for future breakthroughs, as we discussed.
Yeah, and I've been making notes, so I'm probably going to circle back to you at some juncture, because I think it might be helpful for us at the foundation to have a better appreciation for, what are these kinds of testing, who can do them, so we can kind of put a framework together that patients can then take to their physicians and say, hey, did you know that this is available? Here's where you can go, or here's, we can help facilitate some of that collaboration, connection, because I know these tend to be very sensitive and probably very expensive. So we also have to prove to our friends here in the insurance industry why this is most beneficial for them. So that's a whole nother kettle of fish, but for another conversation. But I think also for all of us as patients to ask our physicians if we can donate some of our own samples so that they can be incorporated and tested and run through as part of these preclinical kinds of efforts, right? Because the more samples you have, the more you can understand. So I will encourage everybody to offer their tissue samples or blood samples and things like that to really help with the research. Let's see, I've got some more questions here, so I want to be attentive. Oh my goodness. Let's see, I think we answered your question, Phil, on how to ask your physicians about analyzing and have more personalized treatments. I think we have a ways to go on that. Somebody wanted to ask about, what do you know about Bexarotene? And I'm not sure what that question is related to, whether it's how it works or, the effectiveness. I don't know if you have anything specific to say about Bexarotene and the disease. We've been using that treatment for a very long time.
Yeah, I will say is a couple things. So one, it works very well for many patients, we know. It works very well for patients. Sorry, I think it works very well for patients. Surprisingly, we know very little about how it works. And so unfortunately, because we don't know how it works, we don't know how to predict who it works the best for. But that being said, I'll say is that it affects that first part of the central dogma of how DNA becomes RNA, and it's very effective for many patients with cutaneous lymphoma. But we are trying to figure out, as a field, as well as our lab, is to figure out how it works in lymphoma. It's incompletely understood. And if we knew that, I think we'd better understand who it would work the best for. And so what I'll say is it's one of those retinoids that I said in the DNA to RNA. It works very well for many, many patients, but I think we still need to know better about how it works so we can better predict who should get it and why and how, and also how it can be combined with other treatments. So, yeah,
good point. Lot of things that we still need to know even about treatments that have been around for a long time, because we didn't have that kind of technology to analyze. So more work to be done. Let's see. Okay, someone doesn't have the COVID-19 vaccine. A doctor recommended it. The other does not. Recommends antibodies injections. I think that's always a question, right? COVID and vaccines and how it impacts individuals with cutaneous lymphoma. And I don't know if you have any comments on that.
I do actually. What I can say is that patients with cutaneous lymphoma have actually, we were very worried about them with COVID because we're worried about their immune systems. But it's actually been a remarkable finding that they've done much better than we originally thought. Because your immune system is off, especially when you have these full body rashes. But that being said, we also know that patients with any kind of blood tumor or lymphoma actually do slightly worse than the general population. So in general, I can't speak to individual patients because many patients are doing very, very well with limited disease. But in general, our patients with cutaneous lymphoma should be up to date with their vaccinations, and they would be potentially eligible for prophylactic therapies like antibodies because their immune system is maybe a little bit not as good as the average person without lymphoma.
Great. And I know that there's work being done internationally to collect all of the clinical experience with COVID and cutaneous lymphoma so that an analysis can really be done on the impacts and on the different disease variants and so forth. So we're keeping an eye on that work as well, and we'll share that as things come to fruition. Let's see. You did a great job making highly technical subject understandable. This gentleman is stage 1A and doesn't have any tumors and doesn't qualify for clinical trials. I guess I beg to differ a little bit, right? So maybe you can talk a little bit about who might be eligible for the clinical trials, how these kinds of things work, because I think everyone with cutaneous lymphoma could participate in some way, whether it may be not in a therapeutic clinical trial, but perhaps in some of the research you were talking about, because his lesions or biopsies could be beneficial.
Well, what I would say is that we know that patients with stage 1A do very well. That being said, patients from stage 2 started somewhere. And so we, as a field, are always looking for therapies for patients with all stages. Clearly, the focus has been on late-stage disease to get better treatments. But that's why I said the first statement is our goal is to cure CTCL and CBCL, cutaneous lymphomas. And I'm sure everyone has been told by their doctors that we can't cure even stage 1A disease. We can effectively manage it. You may end up going to complete remission, but nothing that we do cures the disease on its own. So I do think that through research that we are actually actively working on different therapies that may be curative for patients with stage 1 lymphoma. And you may not feel the need to participate in a clinical trial because you're doing so well, but there are periodic clinical trials, especially with skin-directed therapies for patients with early stage disease as well. So why do they occur? They occur because even though our management is really good, even though patients do really well, there is still room for us to do even better. And our goal is to really cure patients where they can undergo treatment and potentially never worry about it again. So you're right. Most of the clinical trial efforts are patients with late stage disease, but there are efforts both in the laboratory, preclinical, as well as in companies to develop treatments that could be better at treating early stage disease.
Great. Gosh, we have a couple more minutes and a couple more questions. So question on CAR T being successfully used to treat and cure other lymphomas, as you had mentioned, particularly on the B cell side. Can you talk in a little more detail about progress and roadblocks for using this in cutaneous lymphomas? It's probably a longer question. We might have to do a whole follow-up on that.
Yeah. So I would say is actually we've made CAR Ts, not we, but the field has made CAR Ts to target all B cells, and that has also cured B cell lymphomas. We cannot make CAR Ts that target all T cells because you need T cells, unlike you don't really need your B cells for many different reasons. But so there's a challenge because how do you only kill the lymphoma T cells but keep your regular T cells safe and healthy? So that has been question number one. Question number two is the CAR T cells themselves are T cells. So without special engineering, the CAR Ts could kill themselves. It'd be a sort of a suicide mission. So for CAR Ts, for T cell diseases, they've had to make special engineering to try to make them so they wouldn't kill each other. So that being said, there's been amazing proof of concepts from multiple sites that suggest that CAR Ts can be effective for at least a subset of patients. I think this will be a major effort at local and national efforts, to try to develop CAR Ts for different types of T cell lymphomas. But I think one of the things where we'll run into a potential issue, as you can imagine, is there's some patients who are N of ones, a cutaneous lymphoma, which has very special features, and they may benefit from a specific type of CAR T. So then how do we make sure that those get developed and actually brought into practice? And so I think what we'll find also, the last roadblock is the relative rarity of these diseases. And so I think patients and foundations have to advocate at every level to try to ensure that there's continued ability to develop CAR Ts, even though the market of patients is relatively small. And so what I would say is that's the final hurdle, which is that we need to continue to use our taxpayer money as well as other research sources to enable, to help pharmaceutical companies develop CAR Ts. But also, I think there's a future where university-based CAR Ts will need to be developed because of the limited market size. But we still need these treatments for some patients. That being said, these treatments are likely going to be effective. We just need a special engineering and proof of concept in clinical trials. And we're very excited to see how and where they'll fit into our clinical therapeutic regimens.
That's a very succinct answer to the question. And I know we're at our limit. I'll ask, and I think, John, your question on how it affects the thyroid, I would suggest ask that question at the open Q&A later on today, because that's a great question for that. And Susan asks about finding a research facility to participate and to send a biopsy to. I think that's a question I'll circle back to you on Dr. Choi to create maybe this guide for patients, right? Where can you talk, how do you talk to your physician, or where can you go if you want to participate and you want to be in, where are some of these research centers like Northwestern, like Dana-Farber, and some of the others that are doing this kind of work. So stay tuned for that, Susan. I think we raised some follow-up work that we have to do. And let's see. Oh, someone really liked your analogies with the smoothies and the visualizations. So very helpful. I thought that was terrific myself. It's like, oh, that's brilliant. So thank you. Yeah, people really want to participate in the research. So I think having a way to send patients and figure out how we can support your efforts is next on our list. So I think we're out of time and we're ready to roll to the next session, or I think we're going into a break at this point. So thanks everybody for your questions, for participating. Thank you, Dr. Choi. This was amazing and brilliant, and really excited to see where we go from here. I have a feeling we're going to have to bring you back a couple of different times so you can update us on how things are going and new and exciting opportunities.
It's such a pleasure, and I love working with you and with all of our patients, and we'll just continue to work more. So thank you very much. I have to run as well.
Yes, great. Thank you so much.